My name is Missy, and this is a presentation that I had prepared for a biochemistry seminar course. This particular presentation was designed to be approximately 30 minutes long and to include multiple research studies in order to convey an idea that is in the areas covered by the field of biochemistry. My presentation, entitled The Neuroprotectivity of Cannabinoids, was intended to introduce the medical efficacy of phytocannabinoids in treating neurological disorders via different molecular mechanisms of action at various target receptors. I can admit it, I was a dare kid, raised during the war on drugs. Man, is pot bad. Marijuana is the gateway drug. If any of us ever tries weed, we'll end up homeless, acting like depraved criminals. You know what? I believed it, as most people did and many still do. But I'm not here to discuss the semantics behind all of that. I'm here to address the importance of cannabinoids in medical science. We'll start with a little bit of background on cannabinoids and why we like them for medicine, and then we'll lead into some neuromedical research. Everyone has heard about ganja, or more properly termed cannabis, but what do you actually know? Cannabis contains more than 100 different previously known phytocannabinoids or natural cannabinoids, with at least 16 new ones being classified recently. Only a select few of these have been studied. These are the cannabinoids that are found in the highest concentrations. The three shaded compounds will be discussed today. We like to study these for medicinal usage for a ton of different reasons, and I'm going to talk about a couple of these reasons. For many neurological applications, their efficacy can be largely attributed to the compound's properties of hydrophobicity. They are nonpolar at biological pH, which is going to allow them to pass readily through the blood-brain barrier, where they can access many of their target molecules. It is also important to note that humans come equipped with an endocannabinoid system, consisting of two major cannabinoid receptors, CB1 and CB2. And we produce endogenous cannabinoids and have enzymes that actively regulate the levels of these cannabinoids under both normal and diseased conditions. While the target molecules of cannabinoids do include CB1 and CB2, the affinity for these receptors is actually considerably low, and they more regularly target different biomolecules in the body. These target molecules include many other types of receptors, ion channels, enzymes, transporters, other proteins, neurons, glial cells, and neural progenitor cells. There is no known lethal dose for cannabinoids. To date, an LD50 has only been theorized, and that is at ratios of 1 to 20,000 to 1 to 40,000. So that would be something like 75 grams of a purified cannabinoid, give or take, and we're not really capable of inducing this response. As with any drug, there is potential for addiction. Here you can see some properties considered in evaluating addiction potential. You can see clearly that it is the least addictive substance on the graph, being compared to nicotine, heroin, cocaine, alcohol, and caffeine. It has low dependence issues, withdrawal symptoms, and tolerance levels, with a moderate reinforcement potential likely due to the intoxication effects of THC. Cannabinoids have therapeutic potential for a wide variation of biochemical issues and perform bioremediative activities in many locations. In the central nervous system, there are applications in adult and neonatal ischemia, brain trauma, Alzheimer's disease, Parkinson's disease, Huntington's, epilepsy, amyotrophic lateral sclerosis, and many more. Today we'll look at some of the neuroprotective actions of cannabinoids. Today we're going to talk about 3 neuromedical research articles demonstrating first that cannabidiol, or CBD, and cannabidivarin, or CBDV, have potential in the treatment of neuronal hyperexcitability, or epilepsies. This was a collaborative research study involving experts in biomolecular chemistry, Pharmacy, and Health Sciences from Pozzoli and Catanzaro, Italy, and Reading, UK. We'll follow this with a discussion of how delta-9 tetrahydrocannabinol, or THC, and cannabidiol, CBD, in combination are neuroprotective, and we'll wrap up with a discussion of how cannabidiol encourages neuritogenesis. Epilepsies are partially caused by defects in membrane excitability, which leads to the aberrant synchronization of neural networks that is characterized by seizures. Epilepsy is the most common neurological disorder, and there's yet to be a cure. Roughly 70% who suffer with epilepsy do achieve symptomatic control with current AEDs, but this leaves 30% with no effective treatment. CBD and CBDV exert anticonvulsant activity. The mechanisms by which these effects are produced are thought to be different from AEDs and are not fully understood. It is noted, however, that one mechanism of action involves triggering TRPV1 mediated intracellular calcium elevation. TRP channels are expressed throughout the body. Their activation transduces chemical or physical stimuli into nerve impulses to then transmit to the brain. They're characterized by weak voltage sensitivity and are non-selectively permeable to both monovalent and divalent cations. This is what makes them a prime target in the treatment of neuronal hyperexcitability. It's important to note that due to the time constraint of the assignment and the amount of information that I wanted to cover, the deactivation was not covered here, though the results were also supportive. The experimentalists aimed to study the effects of CBD and CBDV on TRPV1 and TRPV2 channel activation and deactivation in an in vitro model of epileptiform activity that was modeled in HEC293 cells. A patch clamp assay was performed by evoking a bidirectional current ranging from -80 to +80 millivolts and recording the subsequent charges received. The measurements were then used to determine current density, which is directly proportional to the activation of TRP ion channels. Patch clamp analysis was performed first to investigate activation of the TRPV1 channels in cells that were transiently overexpressing these channels. Capsaicin, also CAPS, is a compound that is found in chili peppers and a known selective agonist of TRPV1 channels. It was used as the positive comparison, and DMSO was the vehicle that was used for negative comparison. Graph A shows the increase in current density with capsaicin at 1 micromolar and 10 micromolar concentrations, indicating an increase in the activation of TRP channels, with the DMSO vehicle that shows no increase in activation as expected. Graph B shows a dose-dependent increase in channel activation in response to CBDV here, with 30 micromolar giving a larger response, though 10 micromolar dosages still activate the channels. The bar graph D corresponds to this graph and indicates the calculated current density, which is another way of showing this increase in activation, and moving from the lower left to the upper left can allow us to visualize this change. Graph C is going to show the dose-dependent increase in activation occurring with CBD as well. This bar graph E corresponds to C and shows a very similar trend, the current density increasing from the lower left to the upper left, allowing us to visualize the increase in activation again. Capzazepine, or CAPZ, is an antagonist that is specific to TRPV1 channels, and it was used as an inhibitor to ensure that the effect was due to TRPV1 activation. So how they ran this was with DMSO as the vehicle, CAPZ alone, CBDV alone, and CBDV plus CAPZ. Then they repeated the same thing with CBD. In figure F, we can see that CBDV will show an increase alone, but shows no difference in the presence of the inhibitor, CAPZ. This is shown again below in the bar graph of the calculated current densities, the increase in activation with CBDV alone and inhibition in the presence of CAPZ. On the right, CBD provided similar results. In graph G, we see the increase with CBD alone and inhibition again in the presence of CAPZ, which is also represented further in the bar graph underneath, the increase of activation with CBD and inhibition with the addition of CAPZ. So this shows us that it was the TRPV1 receptor. Patch clamp analysis was utilized again to investigate the activation of the TRPV2 channels in cells that were overexpressing these channels. Graph A shows a noticeable dose-dependent increase in activation in response to CBDV, but both doses maintain the ability to activate these receptors as well. Bar graph C corresponds to this graph and shows the increase in activation. Graph B shows the dose-dependent increase in activation occurring again with CBD as well, and bar graph D is going to correspond to this and show the activation again. Ruthenium red was the specific inhibitor used to ensure that the effect in this case was due to TRPV2 channels. This was carried out much the same as before, with DMSO as the vehicle, ruthenium red or RR alone, CBDV alone, and CBDV plus RR. And again, it was repeated with CBD as it was for the TRPV1 channels. Figure E shows that CBDV will increase channel activation alone, but will not activate the channels in the presence of RR. This can be seen again below in the bar graph of the calculated current densities. There's a typo down here, but the increase can be seen with CBDV alone and inhibition here in the presence of RR. CBD on the right shows the same trend. In graph F, the increase can be seen with CBD alone and inhibition in the presence of RR, and shown again in the bar graph below, we see the increase with CBD and inhibition with RR. So this was the TRPV2 channel receptor. Microelectrode array is a method that is used to transmit and translate neural impulses into electrical impulses, which enables a computer to monitor the normalization after electrical impulses are received. Recordings in acute hippocampal brain slices from an in vitro model of epileptiform activity showed that CBDV can suppress epileptiform activity and that CBDV also exhibits high potency at TRPV1 channels. Microelectrode array was used to study the effects of DMSO, capsaicin or CAPS, IRTX or iodoresiniferatoxin, a TRPV1 antagonist, CBDV, and then CBDV and capsaicin, both with IRTX, in native rat hippocampal tissue. They provided an electrical impulse and then monitored the normalization. The burst amplitude was found to be reduced by all compounds, but the most by CAPS, CBDV, and CBDV plus IRTX. The burst duration was determined to be affected only by CBDV, and this was established due to the decrease in duration coupled with a low standard deviation. While the difference doesn't appear to be that large on the graph, it is really significant and it's an important find because it is the only compound yet discovered to shorten burst duration. Burst frequency was greatly reduced by CAPS and CBDV, and a combination of CBDV and IRTX appears to reduce the frequency. However, the error indicates that these two compounds together could be quite detrimental for the people who might fall at the high end of this deviation. This study showed that cannabinoids target TRP receptors as one of their mechanisms of action in controlling neuronal hyperexcitability. CBDV and CBD increase the activation of TRPV1 and TRPV2 channels, and CBDV shortens burst duration, the first compound known to do this. Further studies will allow us to understand more of the mechanisms of action behind cannabinoids and their promising applications in controlling neuronal hyperexcitability. So far, we have discussed that CBD and CBDV have potential in the treatment of neuronal hyperexcitability. Now we will cover how delta-9 tetrahydrocannabinol, or THC, and cannabidiol, CBD, in combination are neuroprotective and have potential in therapeutic treatment of neurodegenerative diseases such as Huntington's. This research was a collaborative effort amongst biochemical and neurochemical experts in Madrid, Spain, Cagliari, Italy, and Aberdeen, UK, and then we'll get to how CBD encourages neuritogenesis. Huntington's disease is caused by a CAG triplet repeat expansion in the Huntington gene, which results in an abnormal polyglutamine tract in the amino-terminal region of the protein. This causes the proteins to aggregate and become toxic for striatal and cortical neurons. There are currently no drugs considered effective in alleviating symptoms or delaying the progression of the disease, which will ultimately lead to an early death. Clinical tests using cannabinoids have demonstrated preservation of the striatal neurons against different cytotoxic stimuli in HD pathogenesis. Sativex, a cannabis-based medicine used for multiple sclerosis, is a combination of THC and CBD and has an appropriate profile for HD because it can activate CB1 and CB2 receptors and also exert antioxidant properties. The experiments performed were designed to evaluate the neuroprotective effects of a one-to-one combination of THC and CBD and to monitor the number of Nissl stain cells, which are a selective and sensitive marker for melanate damage in the striatal parenchyma. The neuroprotection was evaluated by measuring the volume of edema using NMR, the number of Nissl stain cells, the number of fluorojade stain cells, and immunohistochemistry with IBA1 and GFAP, as well as Western blot to assess the expression of a few biomarker proteins. Groups of 5 to 6 rats were used. The control group was administered nothing. The vehicle group was injected stereotaxically with malonate to simulate an HD model of neurodegeneration, and the third group was administered a one-to-one combination of THC and CBD 1 hour prior to and 1 hour post administration of malonate. In this figure, we can see the control with no damage beginning or accruing. In the middle, we see the pronouncement in edema immediately after administration of malonate, increasing rapidly over time. And on the far right, we can see that given the administration of THC and CBD, the edema is not able to progress as quickly. The bar graph on the right shows this same data. The edema volume was also physically measured post-mortem at 48 hours and was shown to be significantly smaller due to the neuroprotectivity of these cannabinoids. Nissl staining was performed on the cells from coronal brain sections in the caudate putamen post-mortem. The Nissl staining allows visualization of surviving neurons, so we want to focus on the purple dots. The control has experienced no damage and thus no loss of neurons. Malonate in the center shows a detrimental reduction, and the neuroprotectivity can be seen on the right with THC and CBD. The bar graph on the right represents this same data with the number of cells counted and shows the significance of the difference here as well. Fluorojade staining was also performed on the cells from coronal brain sections in the caudate putamen. The fluorojade staining enables visualization of the degenerating cells, so we want to see less of these. We're going to be focused on the really bright green spots. As we can clearly see, the degeneration is rampant after administration of malonate, and this degeneration is slowed significantly by THC and CBD. They have included a bar graph again to show the calculated numbers of degenerating cells against the control at 48 hours. The progression of neurodegeneration is accompanied by an activation in reactive astroglia and reactive microglia. These activations have been shown to not merely be symptomatic of, but also be contributory to the progression of neuronal death. Malonate induces the activation of both. IBA1 immunostaining was used to demonstrate the reactive microglia. We can see an extreme increase in microgliosis with the introduction of malonate in comparison with the control, and on the right, with THC and CBD, the amount of microglia is significantly reduced. The bar graph on the right shows the percentage in comparison to the control. GFAP immunostaining was performed in order to assess the reactive astroglia, the green globs that we're looking at. We can see the increase in astrogliosis in the center and an impressive reduction in reactive astroglia with THC and CBD, which is again confirmed by the bar graph on the right that shows the percentage. The combination of phytocannabinoids display neuroprotective properties against growing edema. THC and CBD in combination allow higher cell survival while also decreasing the number suffering degeneration. The combination also reduces astrogliosis and microgliosis, both caused by and contributing to neuronal death. First, we discussed how CBD and CBDV have potential in treatment of neuronal hyperexcitability. Now we just finished how THC and CBD in combination are neuroprotective. And finally, we can talk about how CBD encourages neuritogenesis and has potential in therapeutic treatment of Parkinson's disease and other neurodegenerative diseases such as Alzheimer's and Huntington's. Leading into neuritogenesis, or the generation of neuronal networks, I wanted to mention neurogenesis, or the generation of neurons, because they can be quite interconnected. The underlying processes and mechanisms defining post-developmental neurogenesis in the brain are continuing to be elucidated and are very widely studied today. We have evidence of neurogenesis occurring in the marine hippocampus, lateral ventricles, dentate gyrus, striatum, septum, thalamus, hypothalamus, and the olfactory bulb. Several recent studies have identified a central role of the cannabinoid system in adult neurogenesis, related to the ability of CBDs to target, bind, and bioregulate neurons, glial cells, and neural progenitor cells. Cannabinoids have actions in regulation of neuronal homeostasis and survival, the result of a combination of multifarious effects that enable the preservation, rescue, repair, and/or replacement of neurons and glial cells. The final research article also demonstrates a role for CBDs in neuritogenesis, which is neuronal regeneration or the repair and reformation of neural networks. This research study involving neuritogenesis was completed by experts in pharmaceutical sciences in Sao Paulo, Brazil. Neurite loss and neuronal death are early characteristics of neurodegenerative disorders, and the regeneration of neurons is a hopeful strategy for treatments of these disorders. Neurotrophic factors are the current strategy as they play a major role in neuronal regeneration. However, they are extremely limited in their application due to their inability to diffuse across the blood-brain barrier. Nerve growth factor, or NGF, has been associated with the prevention of neuronal apoptosis, and loss and dysfunction of trophic factors such as NGF seem to be involved in the pathogenesis of Alzheimer's and Parkinson's, and decreases in NGF have been seen in the serum and cerebral spinal fluid of patients with Parkinson's disease. This study was designed to investigate the neurorestorative properties of CBD using PC12 cells that are treated with the neurotoxin MPP+, which is a metabolite of MPTP, a common neurotoxin that is found in pesticides. Toxicity was induced using MPP+, and calorimetric assay was used to evaluate the cellular mitochondrial function based upon the enzymatic reduction of tetrazolium salt. The graph shows the steep decrease in cell viability with the influence of MPP+ after 24 hours of incubation, confirming the neuronal death caused by this neurotoxin. Using the same method to determine cell viability, toxicity was induced with MPP+ in select cell lines and then incubated for 24 hours. CBD was added in varying concentration to 3 of the cell lines with induced toxicity and a cell line with no induced toxicity. The control can be seen for comparison and has no additional compounds. MPP+ diminishes viability by more than 50% in this 24-hour period. CBD does not induce neuronal death, but can be shown in all 3 cell lines with MPP+ to significantly increase the number of viable cells remaining. And surprisingly, in this instance, the lowest dose seems to be the most advantageous. To evaluate neurite growth, PC12 cells were incubated for 72 hours. The control had no additional compounds. The cell line in the center was incubated with NGF, or nerve growth factor, and the cell line at the far right was incubated with CBD. The increase was then determined by counting only cells with at least one neurite with length equal to or greater than that of the cell body. And then this was expressed as a percentage of total cells in the field. So we see the calculated increase in bar graph A. Now that they know that CBD encourages differentiation in healthy cells, how about the ones that are suffering from neurotoxic damage and degeneration due to MPP+? Cell lines were incubated for 24 hours with MPP+ and washed, then CBD was added to one of the cell lines and they were incubated again. After this 24-hour incubation, you can see that the newly non-exposed cells have started to regenerate, but just barely. After incubation with CBD, this differentiation was about 5 times that of what was observed over here, which you can confirm by looking at the cell count shown again in the bar graph. Lastly, Western blot analysis was used to determine the protein content of synaptophysin, synapsin, and GAP43. This was done to evaluate the effect of CBD on the expression of the neuronal proteins that are related to synaptogenesis and axonal growth, which are commonly used as markers of neuronal differentiation. There is an obvious increase seen in the concentrations of all of these proteins, demonstrating further the neuronal network regeneration. In commonly used models of Parkinson's disease, cannabinoids have been shown to display neuroprotective properties with an advantage of neuritogenesis. CBDs are quite promising for the treatment of Parkinson's disease and many other neurodegenerative disorders. All right, so first we discussed how CBD and CBDV have potential in treatment of neuronal hyperexcitability or epilepsies. Then we covered how THC and CBD in combination are neuroprotective and have potential in therapeutic treatment of neurodegenerative diseases. We have just finished discussing that CBD encourages neuritogenesis. Cannabinoids have special advantage in neuroprotection, being that their profile is very broad spectrum, demonstrated by their activity at multiple molecular sites, both within and outside of the endocannabinoid system. Cannabinoids will target other biomolecules in need of biochemical modulation. How? We don't know in every case, and these are the mechanisms of action that we are interested in elucidating. So does this mean that cannabinoids have applications in every medical malady, or that it has no side effects? Well, no. There will be some contraindications and side effects, such as with any medicine. But I challenge you to find a drug with more beneficial applications that is as limited in the amount and severity of side effects, with next to no addiction potential and essentially no risk of overdose, that doesn't just control symptoms, but also repairs things. This is a truly remarkable medicine. Thank you for listening. These are the sources for this presentation, and if you're interested, I strongly suggest reading some of the research articles for some of the supportive data that got cut out for time. I hope that you enjoyed it.